This is the biology benchmark corrections for 2019-2020. I apologize for not being able to put them up earlier or sooner. My voice has not been the best, so I've been struggling. But I know that you need them because you need to turn them in by next week when we go back to school. Please make sure you turn them in by Tuesday, since it is Tuesday today. Last day, Thursday. I don't want them on Friday. Okay, so remember that you have to write 3 times the wrong question. Please don't write all of the questions, there are too many. Write the ones you got wrong. So please listen to this lecture so you can understand why the correct answers are correct and why you got the wrong ones wrong. And then please write the correct answer 3 times as well. Not A, B, C, D, the one you should have put. And why you should have put it. Please write I now know 1 time. If there is a picture that I tell you to draw, please draw it as well so you can know what it means. So the first one says, the cell can be classified as a eukaryotic because it. Now remember, you should always highlight the question. In this case, it doesn't have a question, but it tells you what to do. It wants you to classify anything that is eukaryotic. So I have to look at the diagram, right? And what do I see? I see cell membrane, cytoplasm, nucleus. That's important. Who has a nucleus? Eukaryotic cells. I know what? I know that. It's not going to let me write there. Okay. Prokaryotes or prokaryotic cells are what? What are they always going to be? What are they only going to be? Bacteria. And bacteria are very simple. They just have scattered DNA inside. All like that. And they have a flagella and little pili and little ribosomes. But that's it. They have a cell membrane around it and they have a cell wall around it. But they don't have a nucleus or little mitochondria or little lysosome. They do have cytoplasm. They do have jelly in there. So the answers, has a single chromosome? Well, the bacteria does. So I'm going to put a B for bacteria. We have 23 little X's, right? Little squiggles. Lacks a defined nucleus. What does that mean? Lacks. You need to know that lacks means not have. It does not have it. You look at the picture on top. Does it have a nucleus? Yes. So that's not your answer either. Lacks organelles. What are organelles? You know this. I'm going to put little organs. Organelitos chiquitos adentro, right? Organelle in the cell. Those are the mitochondria, the lysosome, the nucleus. So yes, it has it. It doesn't lack it. Has a nucleus? Yes, that is your answer. I now know eukaryotic cells have a nucleus. Okay, and you can pause it if you need to write that down. I'm sorry it's messy, but you can hear me, I hope. Next, in eukaryotes, we just went over eukaryotes. You can go look at the notes on top. This process mainly occurs within the, well, I circled within because that's important. What does that mean? Within, right? A number of cellular processes are involved in the production of energy, ATP, right? Now, what do I know about energy, ATP? I know that it happens in the mitochondria, a little frijolito like that. Yes, the chloroplast, a little frijolito that has little thylakoids. It's usually green. Are you green? No, that's for plants. The nucleus. The nucleus has your nucleic acid. That's not for ATP energy. Endoplasmic reticulum. Remember, you have a smooth and a rough. The rough makes ribosomes. So no, where do you make ATP energy? Mitochondria. I now know ATP energy is in the mighty mitochondria. Ugh, it doesn't fit. All right. Which of the following would be the best evidence? That's important. That a cell is using active transport to move a substance across its cell membrane. What is active transport? We've done this already, guys. Active needs ATP energy. You see the A? You see the A? Active ATP energy. Also, from low to high, remember? Against the gradient. So ATP is being quickly consumed near the cellular membrane. Consume means to comer, to eat. I have no idea what that's talking about. Substances are moving from higher to lower concentrations. That would be, can I write on my test? Yes, so I'm writing an arrow. High to low. Is that what I have here? No. Substances are moving rapidly across the cell membrane. Well, rapid, fast, I guess that might need ATP energy. But I remember speaking about that. Substances are moving through channels in the cell membrane. You know what? I think this one's going to be the ATP one. Even if it says consume, which is kind of interesting. So, I now know ATP energy is an active transport. All living species, including bacteria, use the same energy-carrying molecules to provide energy to cellular processes. Which chemical is this? So again, we're talking about a chemical here. I'm going to have to circle some things on the top because chemical isn't enough to help me find an answer. Now, on the top, if you're confused about number 3, it is A because substances just moving through the channels in the cell membrane can be passive. Remember, what did I write up here? I wrote high to low. I didn't write low to high. I'm sorry, I wrote low to high. I didn't write high to low. So B is wrong. There was nothing that said against. Just to move fast isn't always the answer. ATP would have been the best, even though consumed is kind of interesting to me too. So, all living species, including bacteria, same energy carrying molecules. So, energy carrying, energy, ATP. What's my answer? That one was easy, right? All I have to do is read and circle what's important. I now know energy is ATP in all living species. Did you know that? You know that now. That is in the sentence of number 4. This type of cellular transport is known as, let me see if it tells you anything in the paragraph. Cells often engulf. What does engulf mean? To eat. Remember? Particles or solids from outside of the cell. There it is, outside. Them into the cell by pinching off a section of the cell membrane and surrounding the particles or solid through a vesicle as shown. So do you see how it's going in? If this is out, this is exit. If this is in, this is endo. Inside, adentro. So, Your answer was not osmosis. That's the movement of H2O. Diffusion is spreading of particles. Now, maybe you didn't remember, but does it take energy for you to eat? Um, um. Yes, it does. Does it take energy for you to? Yes, it does. So exocytosis and endocytosis are a form of ATP active transport. You wrote about these in your fluency. So remember, when I tell you write your fluency, read the fluency, it's for a reason, guys. It's so that you learn what these words mean because you will be seeing them again. Shame on you if you got this one wrong. This one was in your fluency, in your 4-a-days. And we even did this with an egg. What's the difference between the egg and the bear? It's the same thing. Which statement can the students confirm? So, B, here is B, is hypotonic. What does that mean? Low tonic. 1, 2, 3. So, water loves to partake. Hypotonic means it has low outside in the solution. It's always talking about the solution. So water is going to go inside. Does that look like water went inside? I think A looks like water went inside, right? It caused the gummy candy to shrivel up due to net water loss. Well, the water did get lost, but hypotonic solution doesn't make sense there. This is probably isotonic. No, I'm sorry. This one is isotonic. Let me erase that. This one that shrunk is probably so as soon as it comes up with hypotonic, hypertonic, isotonic, then that probably means that I should figure out which is which, right? So this one had a lot of tonic in there, so the water went in. I'm sorry. Whoops. There I go again. Well, hyper. So let's fix these bears. Iso means both in and out. Hyper means it's high on the outside, right? So water's going to come out. So there should be a lot of particles there. That's why the bear shrunk. Hypo means it's low on the outside. And it's a lot on the inside. So water's going to go into the bear and make it bigger. So B is hypertonic because it caused the gummy candy to shrivel. You see, that one's the same as the top, but that one is the right answer. B is an isotonic solution because it caused the gummy candy to shrivel up. Would iso cause it to shrivel? No, it will cause it to stay the same. B and C are hypertonic solutions. No, because C, nothing happened to it. So the answer was G, like giraffe. Please make sure you know that hyper means high, lots of tonic or particle. In the solution. It's always talking about the environment, not the cell. Not the egg, not the gummy bear, but where you put it. The solution, right? Where it is at. So, and you need to know that hypo means low particle. Again, in the solution. So after eating a large meal, this happens. Which statement best explains what will most likely happen in the scenario? So I have to look at the scenario. What's happening? You look where it says glucose and blood. What's happening? It looks like there is a low amount there and there is a high amount there. Does it tell me which way it's going? Not really. Does it say ATP energy anywhere? No. Okay. So before you look at the answers, analyze what you have to look at. That way you can decide what you think it might be and not get confused when you see the answers. The glucose concentration will reach equilibrium through osmosis. What is osmosis again? The movement of glucose? No, the movement of H2O. Does it say anything about H2O? The glucose concentration will reach equilibrium through active transport. Again, active transport is ATP, energy, right? Does it say anything about that? No, guys, hello. And what else? It's low to high. So low to high means it's going to go from little to high, right? To more. So that would not be isotonic anyway, or equilibrium. It would be less inside and more outside. You all see that on the picture above? The glucose will diffuse, which means to move or spread, out of the cell due to concentration differences. Well, that would be low to high also. That'd be the same thing as as B. The glucose will diffuse into the cell due to concentration differences. Well, the concentration difference is true. There's low on the inside and there's high on the outside. So this one was true there too. But how is it supposed to move naturally? From low to high or high to low? Right? So D is your answer. I now know that passive diffusion does not need ATP energy. And remember, things usually move so that they can balance. They need to be balanced. And in order for this to balance, the glucose in the blood has to go inside the cell probably, right? Okay, so all of the following are commonalities between viruses and living cells, except, now we haven't spoken much about viruses, but these are some things you need to know. Viruses, not alive. No organelles, because it's not alive. It doesn't have little organs inside. Let me see, they cause disease, some of them. Okay, they can mutate, so they can change, guys, right? Mutate means to change. They do have DNA or RNA. Let's see what the answers say. So we want something that is different. So this is different. This is different. Then again, bacteria also have that, right? No organelles, membrane bound. Okay, viruses have nucleic acids. Would that be a difference? Because it says except, right? Common means similar. So we're looking for something that's not similar. So viruses having nucleic acids, is that similar or not similar? We're going to say that is similar or common. Viruses have a protein capsid. I know we don't have that. Viruses undergo evolution. I think we do too. Viruses mutate. I think we do too. So G's your answer. I now know viruses have a protein coat. And we don't. The protein coat protects the viruses, guys. Shame on you, though, too. Even if you didn't know much about viruses, you should have known that we have nucleic acids. We undergo evolution and we mutate. Protein capsid, you know protein, but a capsid, what? That one should have been the answer to you. So which of the following traits is shared by both bacteria, virus? Both are pathogenic and will cause disease. Even if you don't know what pathogen means, which means causing diseases, it tells you there, causes diseases. So yeah, that is pretty true. They both can cause diseases, right? Bacteria too, you know that. Both contain genetic information such as DNA or RNA. Hey, that's pretty true too. Both require a host cell to reproduce. Bacteria don't. Remember I told you bacteria make copy, 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 copy? I've told you before, copy, 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 or bacteria. So that's not true. Both can be killed with antibiotics. Well, can be killed is a bacteria because a virus cannot be killed, right? It's not alive. Only things that are alive can be killed. Okay. So the answer could be A or B. And we're going to go with the best one. We're going to go with B like boy because not all bacteria cause diseases, guys, although they can. Some of them get you sick, but some of them you have in your intestines. If you ate breakfast, they're helping you right now break down your food. Bacteria can also help you make wine and cheese. So not all bacteria are bad. I now know both bacteria and viruses have DNA or RNA. And that some bacteria are beneficial. Beneficial means good. They benefit you or help you in some way. Which of the following is true about viruses and bacteria? We just did a lot on the top. You can go to the top. You can go to the other one before that. So viruses are much larger than bacteria. Well, if this is a bacteria, by the way, it tells you on the top. This is a virus. It's like a little spider. So who's bigger? Viruses are prokaryotic and bacteria are eukaryotic. Are bacteria like you? No, remember they are prokaryotes and they're always going to be and they're only going to be prokaryotes. Nothing else. So viruses are not prokaryotes. Most bacteria have a cell wall and viruses do not have a cell wall. Well, I think you have to be a cell to have a cell wall. Is a bacteria a cell? What are cells? They're alive. Can you kill a bacteria? Yes, guys, a bacteria is alive. Prokaryotic cells, eukaryotic cells. Bacteria have cells. Are viruses cells? Are they alive? No. So then that is true. Bacteria contain nucleic acids and viruses do not. Well, nucleic acids, didn't we just say in the last one that they both have DNA or RNA? Hello. So then the answer is H. Please know that bacteria are cells and viruses are not cells. Which of the following is most likely the function of the envelope protein? Okay, I'm so sorry you're hearing water. If you're hearing it, I am in the restroom because my kids are still asleep. I don't want them to hear me. I'm not on it though, don't be gross. So, which of the following is most likely the function of the envelope protein surrounding the viral structure? So, here's the envelope protein on the outside. And what do you think it does? It probably, what do you think it does if it's out there? It looks like little sticks, right? Maybe they'll stick to something. What else? Maybe they protect it some way, right? I mean, I don't know, right? Let's find out. So, what you can do is, what do you know? It says we can put protect, we can put stick, we can write on this test to help attach. Okay, yeah, so we said stick. That means attach, right? To create a barrier and protect it from the environmental elements. That sounds pretty good too, all right? Barrier, protect. To preserve the genetic material for future generations. Wait, that sounds the same. To preserve means to keep, but it's like protecting, right? So that sounds like the same thing. To provide rigidity to the viral structure. Okay, well, the best answer actually is A, like apple. I now know viruses need to attach to a host. Remember how I said they're not alive? Well, they're not alive because they cannot reproduce on their own. They cannot make copy like a bacteria. They don't have sperm and egg either. So then they have to need to get to a host, which is a cell, like a bacteria or you, so that it can make more. But it doesn't make it, your cells do. Okay, next one. This type of viral reproduction is known as. So viral reproduction means it's going to make more. And it says a certain type of bacteriophage, that's a virus that attacks bacteria, replicates, that means reproduces, by incorporating its own genetic material into the host genome, where it is passed on to subsequent generations over time. Binary fission, budding, lysogenic cycle, and the lytic cycle. Well, binary fission is bacteria. Is it talking about the bacteria? It's talking about the virus, right? Budding is like mitosis, but that's kind of like what a potato would do. So I'm going to put there plant. Lysogenic is lysogenic. Lytic is fast. Doesn't it sound fast? Say it. Lytic. Lytic. Say that 5 times, right? Okay, so it says here generation over time. So which one would it be? Obviously, we said it wasn't F or G. It has to be H or J because those are for viruses. Yeah, the slow one, right? Duh. So the lysogenic cycle. I now know. Please write it because I can't. Lysogenic cycle is slow replication, and lytic cycle is fast replication in viruses. And I will tell you more about that later. We didn't skip any, did we? No. DNA replication. Oh, I remember that. Do you remember that? We start off, whoops. Well, let me do that again. Remember we start off like that? And then we end up with a half new and half we keep it original. So a new DNA molecule that contains 2 new strands. No. A new DNA molecule that contains 2 old strands. Obviously, it had to do with the new and an old, right? A new DNA molecule that contains 1 old strand and 1 new strand. Yeah, I think C is the answer. An almost perfect copy of the new DNA molecule. What? It has to be a complete copy. What do you mean almost perfect? That's a mutation. You have a disease. It's just a new one and an old one, but it's still the same copy, guys. It's a replication, right? So I now know, and I could have written here copy, right? If you can hear my kids in the background, I'm sorry. I got out of the restroom. I now know DNA replication makes a new copy and is semi-conservative. I can't write it here. Semi-conservative. It means that it keeps half of the original. Sorry. In DNA replication, the nucleobase base, right? I know what bases are, right? A, T, G, C of cytosine always pairs with, oh, I know that. I just wrote it right here. And what does C pair with? Adenine, thymine, guanine, or uracil. Wait, uracil is with RNA, not even DNA, right? So, there it is. I now know cytosine pairs with guanine. Oh, please know that. They're curvy. G and C are curvy. Your gato is your cat. The car's in the garage. Okay. The pentagons. What are pentagons? Do you not know what pentagons are? The pentagons connected by a P. Where's the P? There's my P. Represent which component of DNA? Okay, are you talking about the P or the pentagons? The pentagons connected by a P. The pentagons connected by a P. One more time. The pentagons. So what are the pentagons? Phosphate is probably that P, right? And then sugar. And then the base. I know it's all about the base. I know the bases are here. So then what is the pentagon? The deoxyribose sugar. The sugar. I now know the pentagon is a sugar. I didn't put deoxyribose because in RNA it's not deoxyribose. In RNA it is ribose. Phosphate sugar base. The DNA molecule contains the genetic instructions for an organism. You know that. What are the instructions for a specific organism determined by? What are the instructions? Let me see. In DNA, it's all about the base. And the base has a nucleotide, phosphate, sugar, base, ATGC, right? So I know all of this. The sequence means pattern, of the bases. Oh my gosh. The length, which is how long the DNA is, the type of the bases in the DNA molecule. But there are only 4 types, right? ATGC. Wouldn't that be like the pattern also? The number of nucleic acids. It doesn't have to do with the number, guys. So I now know it's all about the base pattern or the sequence. I'm writing it. It's all about the base pattern. Base sequence. I'm going to write it down. Sequence. So how is the information? Information is important. Carried by DNA into a code. What was the code again? Remember the code were the bases, right? A, T, G, C, A, T. You kept doing that. So I know I'm going to need A, T, G, C, which are bases, right? Okay. So this is the code. By the number of separate strands of DNA, remember these are strands. Does it have to do with how many I have? I thought I only had 2. Why do I have so many now? By the size of a particular chromosome. Remember, your karyotype has different chromosome pairs, but the size really right now doesn't say much. Only the bases that are in the chromosomes. By the number of bases in a strand of DNA. Hey, it says bases. That could be my answer. But number, you mean I only have to have 20 A's and 30 C's? Did Mrs. Martinez ever talk about certain numbers? Maybe in different species, right, of animals? By a specific nucleotide sequence of the DNA molecule. What's a nucleotide? What's that? Didn't we just do that, guys? Phosphate, sugar, base. That's a nucleotide. Say it with me. Phosphate, sugar, base. Nucleotide. Phosphate, sugar, base. Nucleotide. I want to make you say that in class if you don't learn it. Sequence. Hey, that's a word we already used, right? Sequence means pattern. Does the pattern matter? Yes, guys, that's the most important thing, the pattern of the base. The sequence, the pattern of a nucleotide is important. Now, again, in your fluency, you wrote the nucleotide. How many nucleotides are there? Name the 4 nucleotides. You did that in your fluency. It's not just for a grade. You need to know these things. So when you do fluency, please listen and do the work correctly. Which of the following is not a component of DNA? Deoxyribose sugar. I think that's there. Monosaccharide. What's that? Sugar, remember? And then phosphate. I know the phosphate is right there. And the base is right there. So you know your answer already. I now know a nucleotide of a nucleic acid. That's DNA. You see the N and the A? DNA. Nucleotide is phosphate, sugar, base. The arrow in the diagram is pointing to what part of the DNA strand? I see it pointing to P. How about you? So what is that? Is that a base? No, the bases are here. A, T, C, and G, right? The bases. Sugar? I thought I said that the pentagon was the sugar, right? Phosphate? Well, I think it's a P, right? Hydrogen bond? The hydrogen bond would be here, connecting the A with the T. Okay? So, I now know the P in a nucleotide is a phosphate. And that's what it looks like. Which of the following nitrogenous bases will not be found in the DNA of this organism? It says E. coli, that's a bacteria by the way, is a normally harmless bacteria that can sometimes cause serious digestive system infections in humans. So, DNA. It says DNA. I didn't even have to read that if I didn't want to. Which one is not in DNA? I now know uracil is in RNA, not DNA. Which of the following molecules supplies the main source of energy? ATP, used in cellular respiration. So, what gives you the most energy? What are enzymes again? They're helpers, remember? They're proteins. What are carbohydrates? Carbs are sugar. And what are lipids? Fats and proteins. Well, they're also proteins, right? So I knew it wasn't A or D. The answer is B or C. So, main source of energy. It doesn't say the most energy. Main source. What gives you energy, guys? Is it sugar or fat? I now know carbohydrates gives me quick energy. It's the main source of energy. When you don't eat, when you get on a diet and you're not eating carbohydrates, carbs, right? You all have that keto diet now? You don't eat carbs. So then you're not eating your sugar. So then what are you eating? Your body starts to use the fat as energy. And that's why you lose weight. Because you need energy in your body. Hello? And if you're not eating it, then it's going to take it from your body. And then where? From your protein. So be careful with those diets, guys. Which of the following would best match the biomolecule with its function? Biomolecule function, right? Long-term energy. Insulation. That keeps you warm. You know that. What keeps you warm? Fat. So that is lipid, because that's a fat. Provides short-term energy. Didn't we just say that quick energy is carbs or sugar? Help catalyze reactions and build muscle. Muscle. Catalyze, which means speed up. Remember, those are proteins or enzymes. Proteins build muscle. Provide genetic blueprint. I know what genetic blueprint is, DNA. So that's nucleic acid. Do you see the N and the A there? Okay, so W is nucleic acid. No, that's lipid. W is lipid. Yes. X is carbohydrate. Yes. Y is protein. Yeah. Nucleic acid is Z. Yes. Very good. I now know. Okay, it's going to be kind of long. Lipid gives, let's do the long term. Longhas, right? For long-term energy. They keep you warm too. Comma. Carbs or carbohydrates give me quick energy. Short term. If I don't eat that though, then it's going to go into my fat. Proteins are enzymes. They help you quick, right? Quicken that chemical chain that's happening, a reaction that's happening in all of your body right now. Okay? From you breaking down food and all that stuff. Okay, and genetic material or blueprints is a nucleic acid, right? The DNA. Okay. Next. Which of the following correctly completes the table? Adenine, guanine, thymine, and cytosine bond nucleic acids together. So, yeah, A goes with T, G goes with C. What are these? These are bases, right? Nucleotide bases. Okay, so then what are the answers? Store energy used by a cell. That's kind of just ATP. The building blocks of DNA. That's a nucleotide, okay. Can bond with any base pair. No, A has to go with T and G has to go with C. So I don't think so. Is it a type of fatty acid? Is it a lipid? No. So the best answer would be B probably, right? So I now know nucleotides are 4 bases. A, T, G, C in DNA. You need to know, it was in your fluency. Nucleotides are 4 bases. Phosphate sugar A, phosphate sugar T, phosphate sugar G, and phosphate sugar C. That's a nucleotide. That's DNA. Next, which of the following biomolecules, another biomolecule, is responsible for fueling? What is fueling? Usually fueling is like energy. You fuel when you pump gas, right? It gives the car energy. Taylor reactions and structural support. Support inside the cell wall. Well, if this was a cell wall, what's after a cell wall? What is a cell wall protecting? I would say the cell membrane. That would be like a lipid or carbohydrate, right? Hmm. So, is it in the cell wall? Because lipids and carbohydrates are also, the cell wall will be composed of carbohydrates, guys. The cell membrane will be composed of. So remember we said inside, we're going to go with what is in the what is in the cell wall. And in the cell wall is carbohydrates. In the cell membrane would be the lipid. Okay? Now you know. Are in the cell wall. Okay, moving on. Besides solar energy, right? So the sun, what other reactants, which means start with, are needed to ensure photosynthesis occurs? What does photosynthesis need? What does a plant need? Right? Here is a plant. It needs, does it need oxygen? No, you do. It needs carbon dioxide. What else does a plant need? Water. So C was your answer. I now know plants need CO2 and H2O besides the light, right? Which of the following suggests that the pH of an enzyme determines the enzyme's function? So, what is pH? A lot of you got this wrong. pH, I made you write this in your notes. And yeah, some of you didn't do it. That's why you don't know what it is. Acid and base. 1 through 14. And 7 is in the middle. It's neutral, water. It's not acidic or basic, right? Usually. So, enzymes can be found in all life forms. Does that tell me anything about the pH? Enzymes are activated by neighboring molecules. Does that tell me anything about the pH? Enzymes are specific to a substrate. Hey, that's true. But does that tell me anything about the pH? Enzymes are denatured. What does that mean? Destroyed by H bonds that are changed. H bonds. Could that be the pH? Yes, guys, it is the pH. Percent of hydrogen present is pH. That is what that means, by the way. Now you know. Okay? I now know pH can denature, and we went over that word over and over again. It means to destroy enzymes, which are proteins. Remember I told you that proteins can be denatured or destroyed by heat, right? Like your hair. If you iron your hair a lot, it gets destroyed by heat. What else? pH. I said you cannot dye your hair a lot or it gets destroyed. Which of the following is not correct about the enzymes in this reaction? Cellular respiration is the process by which cells convert glucose into carbon dioxide and water. In this process, energy in the form of a molecule called adenosine triphosphate, that's ATP by the way, or ATP is released. Enzymes play a role in the chemical reaction of cellular respiration. The enzymes act as catalysts to slow down the reaction. A catalyst speeds things up. So is it going to slow things down? So that one's not correct, right? The enzymes are proteins. That is correct. Is that what I'm looking for? I'm looking for not correct. Are you not reading what it says? The enzymes are specific to a substrate. That's also true. Am I looking for that? I'm looking for not correct. The enzymes function best under specific pH and temperature. That's true, right? Because if not, it gets what? Denatured. So wait, what am I looking for again? That's why you highlight the question and you underline or circle what you're looking for. Not correct. So A like apple was your answer. And what did you learn? That catalysts speed things up. Speed reactions, that's reactions up. That's why you know that one is not correct, because it says slow down. Which of the following correctly arranges the levels from smallest to largest? Tissue, atom, molecule, organism. I think an atom is the smallest thing, right? And then it goes to a molecule. Yeah, that's true. And then to a cell. Okay. And a lot of cells make tissue. Yeah. And then organ. Yeah. And then organ system. Okay. And then organ. Yeah. Okay. That's my answer. What? What are you looking at, ma'am? I'm looking at this. Which one is the smallest? The atom, right? So then that's your answer. Is it just my answer because atom is the smallest? No, I actually looked at it. An atom makes molecules, right? What is an atom? Hydrogen is an atom, right? If I put H2O, that's a molecule, right? And then I can put a cell, and then a lot of cells together make a tissue, and then organs like your heart, and then organ system, and then organism. Yeah, me. That's right. So this one would have been wrong because that goes from largest to smallest. And what does it say up here? That's why you have to underline and circle the question, smallest to largest. I now know, and please write it all again. Atom, molecule, cell, tissue, organ. You have to know these guys. The organ system and organism. You have to know the order of this because that's your body. What level of organization is the structure labeled A? What is A? A is a nucleus. So then up here, would it be an atom? Wait, I thought atom was hydrogen. Is a nucleus a hydrogen? Would it be a molecule? H2O. Would it be H2O? I don't think so, right? What about a cell? Is it a cell? No, it's kind of before that. So it's between molecule and cell, right? Because it's inside the cell. So between molecule and cell. I know it's a little organ, right? Organelle. So, hey, look, there it is, organelle. But is that my answer? What is the question asking? What level of organization is the structure labeled A? Yeah, it's an organelle. It is asking me that. Good job. I now know a nucleus is an organelle. Shame on you if you put cell. It's pointing at what's inside, not what's outside. It's not pointing at your body, it's pointing at your heart. Or I should say the brain, right? The nucleus is like the brain. It tells you what to do. What would be the next level? Now it's asking for the next level of organization that follows the structure labeled A. So what is that? Is that a cell or a nucleus, right? Well, let's find out. If it's a cell, what comes after cell again? Can I go back to the top again? Yes, I can. After cell is a tissue and then an organ. So is the next level tissue? Is tissue here? No, so it's probably not a cell. In fact, guys, this whole thing is a cell. Okay, the whole thing is a cell. So that means that A again would be what? What's that little dot inside a cell? It's the nucleus, guys. It's just a different picture. So the nucleus would be what? Organelle. But it says what's the next level? Oh man. Remember how we just said that it was molecule on one side and cell on the other side? And then we said it's somewhere in between. Didn't I just say that? And go back and rewind. And didn't I just say that? And then number 29. So then what is the answer? Cell. What? But I thought we just said it wasn't. Yes, but the next one is a cell after organelle. This is an organelle. It's a nucleus. What is next is a cell. I now know that after an organelle is the cell, right? What's after your heart, your brain, your lungs, or your body, right? Or the system, but that's not here either. So the cell. In this experiment, the organisms in the Petri dish represent. So after 3 days, the bacteria, so it's a bacteria, growth on the Petri dish appear to have slowed. The viruses that have infected the bacteria must be inhibiting their ability to reproduce. Inhibiting, remember that? That means stop. So bacteria are stopping, inhibiting, prohibiting, remember? Put prohibit. Okay, so what do they represent? Are they an organelle? Are they an atom? Are they a molecule or none of the above? Honestly, guys, I don't think it's an organelle. That would be inside. Bacteria don't even have that. An atom, that's smaller than a bacteria. A molecule, that's also smaller. So obviously, it's none of the above. It represents an organism, guys. A bacteria is a cell. So it is an organism, okay, or a cell, because it's alive, it's on its own. I now know a bacteria is a cell and an organism. Right? Well, it does say organism there, but it would be a cell, okay? Because a bacteria is a cell. It's one cell, not like us. That's why we need a microscope to see it. Which of the following examples refutes? What does that mean? Negates, denies. That's what refutes means. The statement above. So the only role of bacteria is cause diseases. So obviously it's something that's against that or that denies that. Staphylococcus aureus, that's a bacteria, causing an inflammatory response. Would that be like a disease? I'm going to put a D for disease. Yeah. So with that, negate the statement above? No, it's saying that it is true. E. coli, another bacteria, causing food breakdown in the small intestine. Is that causing a disease? No, because that could be the answer. E. coli causing a urinary tract infection. That's another disease or a disorder, right? Streptococcus pneumoniae causing consolidation of lung lobe. Well, pneumonia sounds like pneumonia. Have you ever heard of that? A lung lobe? Consolidation? I might not know what consolidation is, but I know pneumonia, and that's like an infection. Right? So food breakdown would be the best thing, and that is true. So I now know some bacteria that break down, it says break down food in me are beneficial. There's that word again. It means that they're good. Buenos, right? Which of the following is an example of how bacteria can be beneficial? Whoa, that means good. Bacteria live in animal guts and help with digestion. Help? Digest? Didn't we just do that on the top? Bacteria are used to create medicines to treat infections. That's true. Bacteria help recycle nitrogen in the soil. Yeah, that's true too. Man, all of the above sound good, right? So I now know bacteria are beneficial in different ways. I'm going to put various. You need to know the word various, which means varios, diferentes ways. And make sure you know them. They help with medicines, they help you digest, and they help recycle dead things in the environment, the soil. Which of the following items used by people are microorganisms used to make every day? Okay, well, microorganisms are like bacteria, I guess, right? I don't know. So, microorganisms are living, microscope, harmful organizers, useful, I think this is beneficial, right? Cause diseases. I don't think people, I mean, well, people might use them for um, biological warfare, which means they cause diseases in a whole lot of people, like the Aztecs, right? And the Spaniards. That's what they did. And they kind of weakened the Aztecs. So, oh, look, it says people here. That's what we want to look for, right? So, people. To make what? Well, to make is very important. So, what do we use them to make? Food and shelter. How can bacteria make shelter? Shelter and medicine. Okay, maybe medicine, maybe food. Oh, there you go. Food and medicine. Heat source. I don't think so. So the answer was medicine and food. I now know bacteria can help us make food and medicine. What kind of food do they make? Yogurt, cheese, wine. Yep, use them to make all of those things. What kind of medicine do they make? Well, they help make the insulin diabetes, guys, and some other medicine. Which of the following characteristics would only be found in the eukaryotic side? So, characteristics eukaryotic. So this is for you. Lacks a nucleus, I think is a prokaryote. I'm putting a P. Enclosed by a cell membrane. Guys, they both have a cell membrane. So P and E, prokaryotic and eukaryotic. Contains a genetic code. They both have DNA. More complex cell with organelles. Which one? Look at the pictures. Which one looks like it has organelitos chiquitos adentro? Obviously the E, right? Look at the B. I mean the prokaryotic, right? The bacteria. I don't think so. It just has like a little squiggly. That's the DNA. That's it. So, only in eukaryotes, D, right? And you should know that. I now know eukaryotic cells. Notice how they're both cells, prokaryotic cells, eukaryotic cells, they're both alive, are more complex and, please write the word and, I don't have any space here, I'm trying to hurry up, and have organelles. These are membrane-bound organelles, guys, remember, because bacteria have ribosomes and those are not membrane-bound organelles. Which conclusion can be made regarding the relationship between both cells? So, A is an animal, B is a plant. Let's look at it. A, chloroplast. Do animals have chloroplast? I know that that is a little frijolito that has thylakoids, right? And they are green. Are we green? Are animals green? No. So I think that one's wrong. A is a plant. Okay, that's right. B is a bacteria. It says lysosome, vacuole. What are those again? Organelitos chiquitos. Do bacteria have that? No. So, both cells are prokaryotic. Both cells are eukaryotic. Well, they both have a vacuole, right? They both have little organelles, right? What are prokaryotes? What are they always going to be? What are they only going to be? Bacteria. So it looks like although B might not be a plant, it might also be a eukaryotic cell like you. So J was your answer, guys. That was not difficult if you knew what an organelle was and that they both have vacuoles, which are organelles. They're little organs, remember, inside of the cell. Bacteria don't have that. I now know eukaryotic cells have organelles like vacuoles. Okay, now you know they were both eukaryotic cells. So which form is represented? We're looking for something that's represented, right? What do you see? I see high and I see low. Time. And then what do I see? Well, it looks equal. So what happened? I think it moved from high to low and became equal. Exocytosis means, I already told you this, exit. Active transport means ATP, energy. What else did we say it meant? We said it meant from low to high. Is that what's happening here? We said it meant against the concentration, which means low to high. Mitosis. Wait, what? Mitosis is cell division, the body cells, remember? And diffusion. What is diffusion? Spreading. You're spreading things. I now know diffusion is natural spreading from high to low concentration. You know what? Write it out. High to low concentration. Okay, let's move on. If the paramecium, which is probably that up there, is then placed in hypertonic environment, which of the following will occur? So if it's hypertonic, that means it has a lot of tonic. And what's going to happen? Well, water loves to party, right? H2O will probably go out. I don't even have to look at what it said. It says the paramecium will burst because too much water will enter the cell. Can you see what I just drew? Equal amounts of water will move in and out. Hypertonic is not isotonic, guys. The paramecium will lose water to its environment and shrink. I think that's what I wrote. The environment will cause a mutation. What? In the DNA. I now know hypertonic, which is what it said up here, right? Is lots of tonic. Hyper is high, hypo is low. Lots of tonic or particles. Where? In the cell? No. In the solution or in the environment. I did say that, I think. Okay. Next, DNA replication again is an important step in the cell cycle. Oh, I know the cell cycle. The cell cycle, I can even draw it, right? Something like that. Right? I have G1, S, G2. All of this is interphase, right? And then this is the M phase, which is PMAP, prophase, metaphase, anaphase, and telophase, and then cytokinesis. All right, so DNA replication results in, okay, I know this. It results in what? I have, that's a really not, there you go. And then I have one, and I have another one, and then I have, couldn't do it. Oh, what I mean, the production of RNA. Am I making RNA with DNA replication? 2 daughter cells. Wait, daughter cells is in the cell cycle, but DNA replication results in, what is DNA replication again? The copy. two sets of DNA? I think so. Then I just draw that. New instructions for the cell. Wait, if it's new instructions, is that a copy? I don't think so. So I now know DNA replication produces two sets of DNA, right? Do I see number 1 and number 2 right there? Yes. So I have to make sure I read what it's asking, right? The question here, it's DNA replication results in, but write everything just in case. All of the following are true of DNA replication, except replication occurs in the 5 to 3 direction. Oh man, this one's tough because it's supposed to go in the smallest number, 3 direction. So that one would not be right. Replication takes place in the vacuole. Oh wait, what? The vacuole, but DNA is in the nucleus, so that one's also not right. Replication takes place in the cytoplasm of prokaryotes. Okay guys, prokaryotes don't have a nucleus, so that's actually true. Replication occurs in the nucleus of eukaryotes. That's true. So I do not know why they put F, but the answer is G, because that one for sure is wrong. I now know DNA is in the nucleus. So where would replication happen then? Well, in the nucleus, because that's where DNA is, right? So which of the following proteins is not necessary during DNA replication? So I don't need it. Helicase. Helicase is an enzyme. Do you remember what helicase does? Please Google helicase. Remember, it unwinds and separates. I did show you those pictures of helicase. DNA polymerase, it's making more DNA. RNA polymerase, do I want to make more RNA? Poly means more. Single strand binding proteins, well, I kind of do want to bind my single strands, right? So the best answer here would be the RNA polymerase. I now know poly means many or more. Do I want many or more RNA when I'm making DNA? No, make sure you know what poly means. Poly, many. That one was not difficult. What is the main challenge the scientist faces? I have to read because that doesn't tell me enough. A scientist has a vial of human DNA and a vial of sheep DNA. He suspects they may have been mislabeled. Uh-oh. That means they're mixed up. And wants to test to verify that the vials are correct. We'll have to test this, right? DNA. Sheep DNA is circular, whereas human DNA is linear. A sheep is a eukaryotic cell. And most eukaryotic cells have DNA like lines. Circular DNA is in bacteria or prokaryotes. So the sheep are closer to us than bacteria, guys. So that's not your answer. The DNA in both species contains the same molecules and code. Oh man, imagine if they had the same code, then we'd probably look the same. Are you a sheep? Human DNA and sheep DNA are significantly different, but impossible to tell apart using modern techniques. I'm not sure. We have pretty good modern techniques, so that one's like a maybe. The sheep's genome is identical to the human genome. So the genome would be like the code, guys, the pattern. So the genome is the pattern. So it probably would not be the pattern. So I'm actually going toward G. I don't like that it said code because that's confusing, but they do have the same molecules, guys. A equals T, G equals C. There's a phosphate, there's a sugar, there's a base, right? So that would be the best answer because the genome would be closer to the code. That's the pattern of the bases of the ATGC. And I think we do have enough technology today to figure it out, right? So I now know animals like sheep and, write the word and, humans have similar, which is like the same, molecules. Okay, so we're similar. Let's keep going. Why can bacteria recognize a human gene and then produce a human protein? Really? It's probably because we have something the same, right? I mean, why else would they do that? So, DNA replication in bacteria and humans is the same. I'm not sure about that one because they have a circular DNA and we have little lines. So, would they be able to do what we do? I don't know. When we make replication, the basic components of DNA are the same in humans and bacteria. Okay, yeah, DNA could be the same in human and bacteria. A equals T, G equals C, phosphate, sugar, base. Bacterial cells and human cells contain the same kind of chromosomes? No. DNA, yes, but the same type? No, we have linear, they have circular. Bacteria cells contain the same organelles as the human cells. Do bacteria have organelles? No, they don't. I now know DNA is phosphate, sugar, base in bacteria too. So we all have similar DNA molecules, guys. How does one gene differ from another? Again, a gene is a section of DNA. It has the phosphate sugar base. It has A equals T and G equals C. So let's look at the answers. The size of the nitrogen bases. The size? Really? Did we ever talk about that? Not really, but I guess G and A are bigger than T and C. I don't know if that matters. The phenotypes of the nitrogen bases. Phenotypes means physical appearance. I don't know if that'd be the same as the size. The type of bonds between the nucleotides, well, they're all the same. They're hydrogen bonds, right? This one has 2 and this one has 3. That's why I keep writing 2 little lines and 3 little lines if you can see them. The sequence of the nitrogen bases. Oh, that sounds familiar, right? So I really don't like the way they worded it, but that is your answer. So let me see if we can go a little down. I now know a gene is different from another gene, right? Because of the sequence, the pattern, guys. The pattern of A, T, G, and C. In which biomolecules again, would these amino acids most likely be found? So, what is in nucleic acid? Nucleotide. Phosphate, sugar, oops, that's like not a good casita. Sugar base. Lipids are fatty acids. Carbohydrates are saccharides, right? Or sugars. So the answer would be proteins. I now know amino acids make up proteins. Which of the following statements best describes the major role of lipids Okay, so what do lipids do? Store energy, right? Longhas for later. Insulate, which means they keep you warm. Protect, right? They help you bounce if you fall. Okay, those are the main things. They are the main structural components of membranes. Oh yeah, they're also in the cell membrane, right? Lipid bilayer, that means fat. They move DNA. What? No. They cause RNA to replicate. What? When have we even spoken about that? They catalyze. No, that's like an enzyme. And an enzyme is a protein. So the answer is F. I now know lipid makes the lipid bilayer cell membrane, right? It's a cell membrane. It's around all of your cells, protecting it. Which of the following correctly describes the chemical reaction for cellular respiration? Right? Cellular respiration. It says here, chemical equation, 6O2 plus CHO, right? Glucose, like chocolate. And there's an arrow, which means it makes or produces carbon dioxide, H2O, and ATP. This is what we're looking at, right? It looks like it's the opposite of photosynthesis, right? It releases energy, and photosynthesis requires it. Energy, right? There's energy. The main function is to break down food for energy, right? Photosynthesis produces it. Chemical reaction, there's nothing there. Oh, that's what we're looking for. But what does photosynthesis say? Because usually photosynthesis is the opposite of cellular respiration. Breaks down food, produces food. Releases, requires, right? And look at the equation. They're opposite. So, carbon dioxide and water combine in presence of sunlight to produce glucose and oxygen. So what would I put over here? I'd probably put oxygen and glucose. Oops, I can't write 6 12 6. Right? Oxygen breaks glucose down to make ATP energy, right? That's what happens in the reaction. So it says glucose is broken down into water, carbon dioxide, and energy. Well, okay, yeah. It does say that I make that here, right? See it on top? That could be it. Carbon dioxide and water combine in the presence of sunlight. Wait, what? Isn't that this? Sunlight? Water is used to convert oxygen to carbon dioxide. Carbon dioxide is absorbed and oxygen is released. I don't know. So the best answer would be A, because that's what it says in the equation. I now know cellular respiration uses glucose and oxygen to to make or produce H2O plus CO2 plus ATP. So the oxygen breaks the glucose down, guys, okay, to make all of that. What are two products that are missing from this equation? It says light, chlorophyll, so I know this probably is a plant. Carbon dioxide, water, and what do they make? Oxygen and probably glucose, right? So that one is actually F. I now know plants use CO2 plus H2O and light energy in photosynthesis. I know this. That's how I was able to figure it out. To produce, and that was my answer, right? To produce O2 plus C6 H12O6. Okay. Shame on you if you got this wrong. How does lactase enzyme work? What is happening? Here is lactase. Oh, look, it combined. And then here is lactase still. And it made lactose change into glucose and galactose. What did it do? It made a reaction occur, right? But it separated it here. So lactase increases the activation energy of the chemical reaction. Lactase uses ATP energy to start the chemical reaction. Did I see ATP anywhere? No, I didn't see it. Do you? Lactase is denatured, which catalyzes the chemical reaction. What does denatured mean? Destroyed. If lactase were destroyed, would it be able to help? Think about it. Lactase speeds up the chemical reaction. Okay, so it's A or D. Now remember, if I am a helper, lactase is a helper. Am I going to increase or decrease that energy you need to start? Are you going to eat me? Are you going to get energy from me? No, guys. We know that enzymes are catalysts, right? We also know that enzymes are helpers. So you should have less energy needed, not more. Are you going to carry me or what if I'm helping you? No, right? So it doesn't increase the energy you need, guys. It decreases the energy you need. So I now know enzymes are catalysts. What are catalysts? Speed up reactions. We already did this. That's reactions. And helpers that decrease activation energy. Energy you need to start. Activate. You're going to decrease it. You're not going to use as much because you don't need it because I'm helping you. What is the main function of these digestive enzymes? So, it says amylase, salivary amylase, starch, maltose products. So from starch, I'm thinking it turns to maltose because this is a product. End with, right? Lipase, fats, fatty acid. It looks like starch and fats are big and these are small, right? Protease, pepsin, protein, peptides. Because starch are a lot of hexagons. Maltose is like one. Fats, well, there are a lot of fats. Fatty acid, well, that's just what makes up the fat, right? Protein and peptide. Peptide are proteins, but they're like what make up the protein. So provide the genetic blueprint for every cell. Wait, what? Isn't that DNA? That's confusing. That's not it. Chemically break down food particles. Well, if it's breaking down, it probably gets it smaller, right? So that one sounds right. Increase the activation energy of the chemical reaction. What did we just say enzymes do? Do they increase the activation energy that is needed to start? No, guys, they decrease it, right? Because they're helpers. Absorb and excrete nutrients as chemical waste. I don't know about waste or absorbing. So G is your answer. I now know some enzymes help. So enzymes help break molecules down. Okay, so I'm finished. I hope you don't have any questions. If you do, please let me know. Don't hesitate. 3 times the question you got wrong, 3 times the correct answer, 1 time I now know.